Hello class, today we're in chapter 3. We're going to be talking about cells. I believe there are 4 parts to this lesson. We're going to do it all in one shot. Feel free to break it up however you need to. So our understanding of cells is expressed through the modern cell theory, and that is sort of a checklist of traits about cells that sort of exclude non-cellular biological entities, but basically gives us our definition of life. So a cell is the structural and functional unit of life, also the smallest unit of life, and can be individual, so single-celled, or a combined activity of all its cells, so multicellular with specializations. The structure and function of cells are complementary. Remember that function often comes from shape, so this isn't unusual for us, but the continuity of life has a cellular basis, and that cells can only arise from other pre-existing cells. And the modern cell theory also prescribes that DNA is the hereditary molecule of life, that cells maintain homeostasis, and that there is metabolism here. And so when you look at the human body, over 250 different types of specialized human cells, various sizes, shapes, and subcellular components leading to different functions. And so here we have a couple examples of those different types of cells. In general, all cells, all human cells, have 3 basic parts: plasma membranes, cytoplasms, and a nucleus. Of course, nucleuses are unique to eukaryotes, and human cells, being animal cells, lack cell walls. Beyond this class, you're going to encounter many different organisms that have cell walls. So here's a sort of very generalized cell with some of the organelles. We're going to go over the organelles in more detail in a little bit. Extracellular materials. So the cells themselves are located usually within some type of ground substance, a extracellular matrix of sorts. Usually it's going to be like a fluid, an ECF. These can be interstitial, so the cells will be based in this fluid. It could be blood plasma if the cell is in the bloodstream or in the lymphatics or something like that. If it's in the CSF, the cerebrospinal fluid, then it could be there as well, although the CSF is usually quite clean. And cellular secretions like saliva, mucus, serous fluid. The extracellular matrix is just a ground substance, essentially acting as a glue to hold the cells together. And so this substance is usually like a mixture of sugar and proteins. Cells have a plasma membrane, and the plasma membrane allows for selective permeability between the inside of the cell and the outside of the cell. There are fluids on both sides of this, though: the intracellular fluid on the inside, the extracellular fluid on the outside. They are similar in composition, but there are slight differences necessary to maintain the gradients, the chemical and electrical gradients within. And so by controlling what enters and leaves the cell, you can actually maintain homeostasis. This membrane is made out of lipids, and it is a lipid bilayer in animal cells. There are exceptions to that outside of the animal kingdom, but we'll just keep it very specific for now. This fluid membrane is constantly in motion. It's very dynamic. It's referred to as a fluid mosaic because it has very many different pieces, many different types of lipids that are kind of flip-flopping and reversing around. And within that are membrane proteins, as well as these surface carbs, these cell surface markers like the glycocalyx. These cells can be held together through different types of cellular junctions. We will go through the different types of junctions in the future here, but here's a picture of a sort of cartoon-like plasma membrane with different types of proteins. You see a transmembrane channel protein, you see anchor proteins, associated integral proteins, transmembrane proteins, etc. So the lipid bilayer is composed of 75% phospholipids. So the phospholipid is kind of unique in that it is like a triglyceride, in that it has a glycerol head, 2 fatty acid tails, And then where the third fatty acid tail would go is usually some type of phosphate group. And so this gives it a kind of dual characteristic in that the head is hydrophilic and the tail is hydrophobic. So the head is polar and the tail is nonpolar. 5% is also glycolipids. So these are going to be the lipids that have been modified with some type of carb on the outer membrane of the surface to serve as a cell surface marker. And then 20% is going to be composed of cholesterol. And so cholesterol is intercalated within the plasma membrane to help increase membrane stability. So here we see the polar head and the nonpolar tail. The cell itself will form this sort of micelle formation within a fluid substance. And within the plasma membrane itself are embedded proteins. These allow for cell communication with the environment. So many of our hormones, for instance, that are water soluble cannot cross a plasma membrane. So how would this hormone then send a signal to within the cell? Well, it has to interact with one of these membrane proteins based on some type of receptor interaction. And so they'll actually make up about half the mass of the plasma membrane and have some type of specialized function. Some of them are freely floating, but some of them are tethered to deeper intracellular structure. And so you have integral proteins and peripheral proteins. The integral proteins, as the name suggests, are integral. They're inserted into the membrane. Most are transmembrane, so they'll actually span the entire membrane and have an intracellular side and an extracellular side. And to do that, you need both a hydrophobic and hydrophilic region. And so these can actually act as channels and carriers. They can act as enzymes, as in catalyzing reactions, and they can react as receptors. The peripheral proteins, on the other hand, are not actually embedded in the membrane, but loosely attached to perhaps even other integral proteins. These have filaments on the intracellular side for plasma membrane support, and they can function as enzymes, motor proteins, and cell-to-cell connections. So here we have a sort of review of the different tasks that many of the membrane proteins perform. Transport is a very important one. There are many different classes and types of molecules that perhaps need help getting across the semi-permeable membrane. A protein channel can allow for facilitated diffusion, allowing those molecules to move down their chemical gradient and follow the chemical gradient without actually using energy. Otherwise, they simply would not be able to diffuse across the plasma membrane. Some of them do actually use ATP, so would be a form of active transport to allow active pumping of substances across the membrane. Some of these are acting as receptors for signal transduction. So a messenger binds, there's some sort of change of shape to that protein, it releases something on the other side or has some action on the intracellular side, resulting in an effect. Some of these catalyze reactions. They have enzymatic activity. Some of these are necessary for cell-to-cell recognition of the adjacent cells, so glycoproteins, for instance. These cell surface markers allow for recognition by other cells. Some of these serves as anchoring proteins, so allow for attachment to the cytoskeleton or the extracellular matrix. Cell-to-cell joining, so these sort of anchoring proteins that allow for adjacent cells to be hooked together. Going on off this idea of using carbs, this is the glycocalyx. It has sugars basically sticking out of the surface of the cell. Some of these sugars are attached to lipids and some are attached to proteins. We call those glycolipids and glycoproteins. And every cell type has a unique pattern of a sugar coating. This allows for specific biological markers, allowing for specific cell-to-cell recognition. This is also how cell types differentiate between self and non-self. So this is my cell versus this is the cell of an invader. Similarly, the invaders are using these cell surface markers to recognize cells that they can take advantage of. So, as mentioned before, junctions. Some cells are not bound to any other cells, such as blood cells or sperm cells. That is just a functional requirement of those cells. But most cells are actually bound together in these sort of functional units. And the way that these cells can actually be bound to each other is a cellular junction, such as a tight junction, a desmosome, or a gap junction. Tight junctions are integral proteins on adjacent cells that are linked together. And they are tight because they prevent fluids in most molecules from moving in between cells. Now think about where these might be useful in the body. Anywhere you would want to sort of sequester an organ or something like that. So the blood-brain barrier, for instance, is protected by tight junctions. The small intestine is, using bacteria to ferment our food and obtain nutrients that otherwise we would not be able to from that food. You need to be able to separate your own bloodstream away from the small intestine. So you have tight junctions there doing that. Desmosomes, on the other hand, are the sort of rivet-shaped junctions formed by linker proteins. We call these cadherins. And these cadherins link neighboring cells like the zippers of a teeth. And so you tend to find these, I believe, in the cardiomyocytes, for instance, of the heart cells. They allow the heart cell to beat as a unit and stay together during contraction. But the keratin filaments here can connect plaques intercellularly for adding anchoring strength, and they allow give between cells, reducing the possibility of tearing under tension. Another good place might be the epithelial, for instance. Gap junctions. Gap junctions allow for the passage of material, as the name implies, gap. They have a certain size hole that allows for passage of nutrients and ions. They're formed by transmembrane proteins like connexons that form tunnels essentially from cell to cell. So the plasma membrane needs to allow for constant exchange of material. Some molecules will pass very easily, some will not, because the plasma membrane is selectively permeable. There are two ways to move across the membrane: passively and actively. In a passive transport, no energy is required. In an active transport, you need to use ATP to power a pump or something like that. So passive, three types of passive: simple, facilitated, and osmosis. These involve simple diffusion, just following the natural concentration gradient. And all molecules truly have movement from their kinetic energy, and this movement results in collisions. The more collisions you have, because of the more molecules, you will scatter to lower concentration energies to areas. It is just a statistical effect. So this is how that works. So the speed of this diffusion can be influenced by three factors: the concentration, the molecular size, and the temperature. So the greater the difference of concentration between two areas, such as high to low, the faster it will diffuse from high to low. Smaller molecules diffuse faster, and higher temps lead to higher kinetic energy, which leads to higher scattering. Eventually you will reach equilibrium. And equilibrium is not no movement, but no net movement. Now, molecules will want to diffuse down their concentration gradient naturally. This is the most statistical way to diffuse, and it is the path with the least resistance, essentially. But the membrane is posing a problem because the membrane is only selectively permeable. If you are nonpolar and hydrophobic, you can actually pass across the membrane very easily. But if you are sort of hydrophobic, or a large molecule, for instance, you will have trouble diffusing even across a membrane, even if there is a concentration gradient. And so simple diffusion would be the best case scenario of either a polar molecule or a small molecule following its gradient through the membrane. If a larger or non-lipid soluble polar molecule crosses with the help of an assistance or carrier molecule like a protein, we would refer to that as facilitated diffusion. Osmosis is a special case. Osmosis is generally the name for a solvent moving. In our case, usually water. It doesn't have to be water, but water usually is referred to as osmotically the diffusion of water, not molecules themselves, not the solutes. So simple diffusion, nonpolar lipid-soluble substances will diffuse directly through the bilayer. That includes small gases like oxygen, carbon dioxide, the steroid hormones because they're lipid-soluble, and the fatty acids. Facilitated diffusion, this will require a protein carrier usually, but contains hydrophobic molecules like glucose, amino acids, and ions. They are moving down the gradient, so even though they have help, they are not using energy. And so this carrier-mediated and channel-mediated. And in carrier-mediated, you have these transmembrane integral proteins transporting specific polar molecules like sugar and amino acids. When the molecule binds, it will cause the carrier to change shape, and as a result of that, it basically opens up to the inside. And the problem is that carriers have a limit, right? And carriers can be saturated when all molecules are bound. If it's channel mediated, the channel will usually have some kind of pore that moves all the way through the membrane, so it is transmembrane, and then the molecule will just follow the flow of water or extracellular fluid or whatever it is. And so this specificity then will be based on the pore size as well as the charge. If it's a water channel, for instance, we call it an aquaporin. And you have two types of these. You have the leakage channel, which will always be open, and a gated channel, which has to be controlled by a signal of some kind, either chemical or electrical. Osmosis. So on the surface, osmosis is simply the diffusion of water. In truth, it is more complicated than that. There are binding effects that have to be taken into account. There are colligative effects that have to be taken into account. It is the movement of a solvent and not the solute, which is kind of counterintuitive perhaps to these other types of transports, but flow occurs when water concentration is different on one side of the membrane. We can measure that with osmolarity, and this will be the number of solute particles in a solvent. And the solute particles tell you essentially that there is more or less water in there as well, because it's your water concentration varying with the number of solute particles. And that itself is kind of hard to think about because it doesn't actually matter how big the solute particles are, it just matters that there's a certain number of them. And so water moves by osmosis from areas of low solute concentration to high solute concentration, and until they reach equilibrium. So this movement of water creates an osmotic potential, which is measurable in the form of hydrostatic pressure and osmotic pressure, but the hydrostatic pressure is exerted on the cell side of the membrane and can be caused by increases in the volume of the cell due to osmosis. The osmotic pressure is the inward pressure due to tendency of the water to be pulled into the cell with higher osmolarities. And you can probably see the problem starting to build up here. The cell needs to maintain a somewhat relative solute concentration to its environment, otherwise it has to deal with all this water. So when hydrostatic pressure equals osmotic pressure, there'll be no net movement of water to the cell. In this case, plant cells, for instance. So plant cells have a strong cell wall to try and limit that hydrostatic pressure, which in turn limits the osmotic pressure. So a plant cell has a maximum limit. It will fill up. The plant cell will actually increase inside until it is swelled inside the cell wall, and it won't lyse. Animal cells, remember, do not have cell walls, therefore we can't limit the hydrostatic pressure, and we will burst. Our cells will burst if we take too much water in. But the opposite is also true. We can lose water from our cells, causing it to shrink and then lyse that way. Call that crenation. So this tonicity then is the ability of a solution to change the shape or tone of the cells by altering the cell's internal water volume. So we have three terms to describe that. If it is isotonic, the osmolarity is the same inside and outside, so volume remains unchanged. If it's hypertonic, it has higher osmolarity inside the cell, so more solute inside the cell. Water flows out, resulting in cell shrinking. This is referred to as crenation. Hypotonics have lower osmolarity than inside the cell, so water will flow in to try and reach equilibrium, and as a result, the cell will lyse. So this osmolarity is equal to the molarity times the number of ions, in this case the particles in general. Notice that there are other things dissolved in water than ions, but for this example, we're using salt, in this case sodium chloride. So one particle of sodium chloride ionizes to sodium and chloride ions. The problem is this isn't exactly how ions actually work in solution, but 1 molar solution of NaCl equals 2 osmolar solution, just because it's dissociating both into the cation and the anion. We express osmolarity in osmos per liter. So if you look what happens to the red blood cell in each of these, the isotonic solution, the red blood cell will maintain its dumbbell shape. In the hypertonic solution, the cell will crenate as the water leaves the cell, and in the hypotonic solution, the water will move into the cell and cause it to burst like a balloon. So moving on to transport. Two types of active transport, active and vesicular. Any type of active transport is requiring energy, usually in the forms of ATP. And in this case, we're moving solutes across the plasma membrane for whatever reason. The solute is too large to actually use a facilitated channel. The solute is not lipid soluble, so it would never diffuse on its own. And the solute, for whatever reason, is not able to move down its own concentration gradient. And so you can use a pump in this instance. And so these bind specifically and reversibly with the substance being pumped. There are different types of pumps, like an antiporter, for instance, transports one substance while transporting a different substance out of the cell. So while one is going in, another is going out. If it's symporters, it will transport two in the same direction. If it's moving against the concentration gradient from low to high, then of course it will require ATP. And there are two types of active transport, both a primary and a secondary. In primary active transport, the energy used is coming directly from ATP hydrolysis. So the ATP is literally there. It's using water to split into ADP and inorganic phosphate, and now that inorganic phosphate will be used to power primary active transport. In secondary active transport, that effect is kind of peripheral. The required energy is obtained indirectly from ionic gradients that are just a result of the primary active transport. So energy from hydrolysis of ATP leads to a change in the transport protein. That's due to the addition of the inorganic phosphate. Now the solutes will bind the protein and be pumped across. Examples of this, the calcium pumps, hydrogen ion pumps, and the sodium potassium pumps, all very important. Sodium potassium pump is the sort of most studied. This is our ideal pump for this, but basically it's an enzyme. It's sodium potassium ATPase. The pumps sodium out of the cell and potassium back in. And this is how we actually maintain our resting membrane potential. All plasma membranes have it, but especially the nerve cells and the muscle cells, the especially excitable cells, have lots of these sodium potassium pumps. We mentioned before these sort of channels that are always sort of open, the leakage channels. In this case, leakage channels are also responsible for maintaining our resting membrane potential. They leak sodium into the cell and leak potassium out of the cell. In this case, both traveling down their concentration gradients. the sodium-potassium pump is working as an antiporter, pumping sodium out of the cell and potassium back into the cell. So the exact opposite of the leakage channel itself. This is what maintains our resting membrane potential and the electrical chemical gradient, which allows us to generate action potentials and excitable muscles and stuff like that. Here's a picture of how that works. So, talking about secondary active transport, we have used the hydrolysis of ATP to pump something across. And with that, we've actually generated another electrical chemical gradient. And depending on that ion gradient, we can actually use it to pump something else indirectly. So if there is a low sodium concentration inside the cell, then the sodium-potassium pump will strengthen the sodium's drive to want to enter that cell, and the sodium can drag other molecules with it as it flows into the cell through the carrier proteins. This is actually how glucose enters the cell. Glucose is following essentially the sodium, certain amino acids, other ions. We call it a secondary active transport because even though it's not the intended target, it can hitch a ride. Vesicular transport. So vesicular transport involves the use of a phospholipid vesicle, and this is used for large particles, macromolecules, and even fluids in some instances in membranous sacs called vesicles. This does require cellular energy. There is endocytosis, transport into the cell, exocytosis, transport out of the cell, transcytosis, which is across possibly and then possibly out of the cell, not always. And endocytosis is broken down into three types: phagocytosis, which is cell eating, pinocytosis, cell drinking, and then receptor-mediated endocytosis, which is some type of hyperspecific endocytosis. So this involves the formation of protein-coated vesicles, usually receptor-triggered, so can be very selective. Some pathogens are able to use this receptor to actually enter our cells. And so once the vesicle is pulled inside, it could be fused with a lysosome for digestion, or it could undergo transcytosis to be transported somewhere else in the cell. And here's how that works. Essentially, there is a specific receptor binding that allows for the endocytosis to occur, and now that material is on the inside. So phagocytosis is cell eating. In this case, pseudopods will form and sort of engulf solid particles, forming a vesicle, and then pull it into the cell. This vesicle is called a phagosome, and then it will be fused with either some type of digestive organelle or something like that. This is how these sort of primitive single-celled organisms eat, and this is how macrophages and white blood cells help to keep our body safe. So phagocytic cells move by this sort of amoeboid motion where they can move kind of with their extensions as the cytoplasm flows into it. So phagocytosis, you're engulfing some type of food particle or pathogen. In this case, they were engulfing a pathogen. Moving on to pinocytosis, or cell drinking. This is fluid phase endocytosis. The plasma membrane will enfold on itself, basically capturing some of the extracellular fluid and all its dissolved solutes, and then bring it inside the cell. And from this point, it will fuse with an endosome. Some cells use this to sample the environment. So this didn't come up so much in the cell theory, but cells need to be able to respond to an environment. And one of the ways to do that is to sample and check for chemical concentrations. That way, if you detect nutrients nearby and you're sampling, then you can get a feel for which direction it's coming from and you can move toward it and go get it. And so this is the main way in which nutrient absorption occurs in the small intestine. And these components are then just simply recycled back. So the cell is essentially gulping a drop of extracellular fluid. So as mentioned before, this process can be very specific. In the case of receptor-mediated, there is a receptor embedded in a clathrin-coated pit. The entire pit basically gets internalized upon activation. And so examples of this, enzymes, low-density lipoproteins, iron, insulin, viruses, diphtheria, cholera toxins take advantage of this. But the caveolae may have smaller pits and different protein coats from clathrin, but can still capture specific molecules. So very specific because it's not just endocytosis. Now it has to be triggered by something specific. Now the opposite is also true. You can bring stuff into the cell, you can send stuff out of the cell. So this is signaled by cell surface receptors or perhaps a change in membrane voltage. And so what happens is the waste material, whatever it is put inside a secretory vesicle. It triggers exocytosis by fusing with the plasma membrane and essentially folding it in such a way that the contents are ejected. And so some substances exocytose to include hormones, neurotransmitters, mucus, and cellular waste. And so that's what that looks like. So RMP, resting membrane potential. This is the electrical potential energy kind of upheld by the relationship between different ions like sodium, potassium, chloride. A difference between 2 points is going to be referred to as a voltage, and cells that have a charge are said to be polarized because it's a differential. The voltage is only measurable at the membrane surface. The rest of the cell and the extracellular fluid are actually going to be neutral. And so these range in value from -50 to -100 millivolts in different cells, but they indicate the inside of a cell is more negative relative to the outside of a cell. Potassium is the key player in maintaining your resting membrane potential. And when you think about it, it kind of makes sense, right? Because potassium is one of those that is constantly leaking out of the leakage channels. And then your sodium-potassium pump is just going to pump it back in. The problem is charged ions are not very good at getting across that plasma membrane. And so in this case, negatively charged, for instance, we haven't even given an example of one that is able to do so, but the cytoplasmic side of the cell membrane tends to become more negative because of this bias towards the positive ions. But when you pull potassium back in, the more negative interior, you're driving essentially an electrical gradient. So when the drive for potassium to leave the cell is balanced by its drive to stay, that's when you get a resting membrane potential. So it's the electrical chemical gradient of potassium that sets the resting membrane potential, even though you do have other ions at play here, chloride ions, which are negatively charged, sodium ions, which you're pumping more of, etc. So in many other cells, as I mentioned, the sodium is also very important. Sodium is attracted to the inside of the cell because of negative charge. Sodium being positively charged is attracted to its opposite. If it enters the cell, it will bring the cell's resting membrane potential up to -70. The thing is, the membrane is more permeable to potassium than sodium. So chloride ions don't influence RMP because its concentration and electrical gradients are exactly balanced. So there's no way for the chloride ion to get across, but it is sort of maintaining this negativity of the outside, of the inside, the cytoplasmic side. So the pump itself, the sodium-potassium pump, is continuously ejecting 3 sodiums and bringing in 2 potassiums. And so this is what is helping to maintain that RMP. Neurons in muscle cells, because of their excitatory nature, are able to upset the steady state by intentionally opening those gated channels. So the cell is constantly interacting with its own environment. It's responding to other cells. It's dealing with extracellular chemicals that are telling it to do these sort of negative interactions or positive interactions. These interactions always involve the cell surface markers of the glycocalyx. So cell adhesion markers like CAMs, plasma membrane receptors, etc. Every cell has thousands of these projecting from their membrane, and their point is to anchor cells to the ECM or to each other. They assist in movement. They signal white blood cells to injured or infected areas. They can simulate synthesis or degradation of the adhesive membrane junctions, and they transmit signals to direct migration, proliferation, and specialization. So these membrane receptor proteins then have several jobs, including contact signaling and chemical signaling, basically whether or not the cell is touching and recognizing another cell's unique surface membrane, or interacting between receptors and ligands. So ligands cause different responses in different cells depending on the pathway that that receptor is actually part of. And so this ligand receptor binding is very specific, but it's also doing the same thing. It's changing shape. And when you change shapes, you change functions. And so these activated receptors then are changing their functions and becoming enzymes or opening and closing ion gates. They're causing changes in excitability. In the case of a G protein linked receptor, they're causing a cellular change by activating a G protein on the intracellular side. This can cause the release of secondary messengers, which will amplify a signal. Moving on to portions of the cell, we have the cytoplasm itself. This is located between the plasma membrane and the nucleus. It's just a gel-like material like cytosol, mostly water, but it has soluble molecules like protein, sugars, and salts. It does have these insoluble molecules in it, inclusions. These can be glycogen granules, pigments, lipid droplets, etc. And it has the organelles, these tiny organs essentially. These are macromolecule assemblies that have a job essentially. So different types of organelles. The membranous kind would be the mitochondria, endoplasmic reticulum, the Golgi, the peroxisome, and the lysosome. Non-membranous would be ribosomes, cytoskeleton, centrioles. So an important distinction here, eukaryotes have membranous, prokaryotes do not. So prokaryotes have some sort of equivalent to these, but none of these specifically. So the mitochondria, know this as the powerhouse of the cell essentially. It's generating ATP via aerobic respiration. It's got a double membrane by itself. The membrane has many folds called cristae, which help increase surface area. And these cristae are embedding with membrane proteins that help serve in the electron transport chain. Mitochondria contain their own DNA, their own RNA, and their ribosomes are actually a different size than ours. This is one hint that it probably came from a prokaryotic precursor. Then we have the ribosomes. The ribosomes are non-membranous. They are essentially macromolecule assemblies made up of protein and ribosomal RNA. It has two different forms in the cell. Free ribosomes synthesize proteins out in the cytoplasm. Membrane-bound ribosomes synthesize proteins that need to be modified by the endoplasmic reticulum. The endoplasmic reticulum is just a series of flattened, parallel, interconnected sacs, essentially. And it has lots of jobs, but there are two different varieties that we like to make a distinction between, rough ER and smooth ER. The rough ER appears rough because it is studded with the ribosomes. Remember, ribosomes are the site of protein synthesis, and if you're synthesizing into these flattened sacs, essentially, you're sending these proteins into the cisterns to be modified within. After that has occurred, after the modification has occurred, you can package it and then send it to the Golgi apparatus for further processing. Now the smooth ER. This is the looped tubules continuous with the rough ER. Very often is actually attached to the rest of the endomembrane system, including the nuclear membrane. But it has enzymes in its plasma membrane that are useful for lipid metabolism, absorption, synthesis, and transport of fats, detoxification of chemicals. It converts glycogen to free glucose. Remember, glycogen is one of those just cellular inclusions. Storage and release of calcium, especially in muscle cells, where we would call it a sarcoplasmic reticulum. And the endoplasmic reticulum is often packaging and sending to a complex called the Golgi apparatus. So the Golgi apparatus is a series of stacked and flattened membrane sacs. It looks very similar to the endoplasmic reticulum, but it's modifying, concentrating, and packaging proteins and lipids received from the rough ER. So some steps involved. The transport vesicles from the ER fuses with the cis face of the Golgi. The proteins are taken inside for their modified, tagged, sorted, and packaged, and then the Golgi will actually send them out of their trans face towards the intended target, either outside of the cell or further in the cell, etc. So depending on its contents, there are three ways the final transport vesicle can go. So a secretory vesicle will send it outside of the cell by fusing it with the plasma membrane and exocytosing it. In pathway B, vessels containing lipids or transmembrane proteins will fuse with the plasma membrane, basically embedding them there. And in pathway C, lysozymes will remain in the cell, holding the contents in the cell until necessary. Okay, moving on to peroxisomes. Peroxisomes are sac-like organelles that contain detoxifying substance, like peroxides. They have free radicals that are extremely toxic, highly reactive. And the two main detoxifiers are oxidase, which uses oxygen to convert toxins to hydrogen peroxide. And then peroxisomes also have catalase, which convert the hydrogen peroxide to water. So this is kind of a two-step process. First, you convert toxins to hydrogen peroxide and then hydrogen peroxide to water. These peroxisomes also play a role in the breakdown and synthesis of fatty acids. Oxidative materials like this are very destructive for macromolecules, so that makes sense. Lysosomes. So, interesting thing about lysosomes, they're sort of unique to us. So you don't find them in plants, for instance, but they're spherical membranous bags that have our digestive enzymes in them. For instance, hydrolases. This is where harmful intracellular pathogens might be sent, for instance. Ingested bacteria, viruses, and toxins will be sent here to be destroyed. This is also how we recycle non-functional organelles. But essentially, you want to break down and release the constituents of these things to be reused later. In the case where a lysosome is lysed, it could cause autolysis, essentially self-digestion. And so this is what they look like at 20,000 times magnification. But you can see where the materials are being digested within by the enzymes. Okay, so the endomembrane system. This is the series of the membranous organelles. Many of them are continuous with one another. They are working together to produce, degrade, store, and export the macromolecules. They also serve as detoxification for chemicals. Moving on to the cytoskeleton. This is a series of fibers, essentially, filaments, that give us structure, give us motility. There are hundreds of different proteins all linked together, associated with this, linked, cross-linked, peripheral, perpendicular, parallel. And so they act essentially as the bones, ligaments, and muscles of the cell. And also cellular highways. This is how organelles get moved by motor proteins. So three types: microfilaments, intermediate filaments, and microtubules, all classed based on their diameter and their composition. So the microfilaments are the thinnest of the cytoskeletal elements and are composed of actin. And so you probably heard of actin before as the muscle protein. Well, actin is also the microfilament protein. So each cell has a unique arrangement but shares a terminal web. And this is just a network of microfilaments, but basically strengthen the cell surface and help to resist compression. Some cells use this for motility and also changing the shape, and can be used to help sort of squeeze off parts of the plasma membrane for endocytosis or exocytosis. Intermediate filaments. As the name implies, the diameter here is intermediate between a microfilament and a microtubule. So these are tough, they're insoluble in water, they're fibrous proteins, essentially. And they're composed of 4 of these fibrils twisting together, resulting in one strong cable-like fiber. And they're essentially helping to resist pulling forces, so attached to plaques and act as guy wires. Depending on the cell type, they can have special names, like neurofilaments versus keratin. So microtubules, these are the largest of the cytoskeleton elements, and they are composed of the protein tubulin, which has a series of subunits. The microtubules assemble by the dimerization of these tubulin subunits, usually radiating from a centrosome area of the cell. These actually help to determine our overall shape and distribution of the organelles. Many of our organelles are actually physically tethered to microtubules to keep them from simply floating off to somewhere where it would be chemically inconvenient. And so these attachment points are actually used by motor proteins, which use the microtubules as tracks to drag the organelles where they need to be. And so the motor proteins are powered by ATP, but basically just drag stuff around the cell as necessary to get their cargo where it needs to be. And so here we see the actual diameter of the microtubule as well as the different subunits. I believe it's an alpha and beta subunit, basically forming this spherical ladder type shape. So the centrosome and the centrioles. The centrosome, when you look at the etymology of the word, it sort of means cell center or cell body. We also call it a MTOC, the microtubule organizing center. It has a granular matrix and it has the centrioles. These are barrel-shaped organelles that lie at right angles to each other. Centrioles do some cool stuff with like motors, cellular motors, and they form the basis of cilia and flagella, the appendages used for cell motility. But microtubules will assemble at the centrosome and radiate out to the rest of the cell. And so during cell division, for instance, they will radiate out from the centrosome until they meet the kinetochore of the chromosome. So here are the centrioles. They lie at right angles to each other. And you can see the kind of arrangements that we're starting to get when we talk about these sort of complex motors. So extending from the cell surface, you also have these sort of finger-like projections or hair-like projections like the cilia and the flagella, which aid in movement. Cilia also help to move materials across the surface of the cell. Microvilli are finger-like projections to help increase surface area, for instance, like absorption in the intestine. Cilia are going to be more whip-like. They're going to work in mass in a sweeping motion to move, for instance, mucus and everything that's entrained in just one direction. Flagella are longer and powerful enough to propel the whole cell. For example, the tail of a sperm. And so flagella have evolved multiple times in history. When you look at eukaryotic and prokaryotic flagella, they look similar but actually work differently. Both structures are made up of microtubules, and those microtubules originate at the centriole themselves, the basal bodies. They have a 9+2 pattern of microtubules, so 9 sets of double tubes surrounding a central pair of doublets, and that's different from the centriole. The cilia has a power stroke and a recovery stroke. producing a current essentially, which helps to move the mucus forward. Okay, microvilli, these are going to be the finger-like extensions of the plasma membrane. You find these in the intestine and the tubule cells of the kidney, and their job is to increase surface area for absorption. They have a core of actin filaments used for stiffening of those projections. And so when you look at this on a slide of like the intestine, we would tend to say it forms a brush border. Okay, moving on, we have the nucleus. The nucleus is the largest organelle, typically the most predominant thing you see in a eukaryotic cell. It contains a genetic library of blueprints, essentially all the hereditary information in the form of DNA. And it responds to the signals that dictate which proteins are actually needed to be synthesized. So most cells are uninucleate, but skeletal muscle, certain bone cells, some liver cells are multinucleate. And some cells are actually anucleate. So the red blood cell, for instance, has no need for nucleus. It needs to hold a lot of hemoglobin, so gets rid of it. Three main structures: the envelope, the nucleoli, and the chromatin. So here's your typical nucleus. Stains very darkly usually. The nuclear envelope is actually composed of a double membrane. It has its own sort of unique cytoplasm here called a nucleoplasm. The outer layer is actually continuous with the rough ER and studded with ribosomes. The inner layer, which is a nuclear lamina, has a mesh of proteins maintaining its shape and acting as a scaffolding for the DNA to work. Entry and exit from the nucleus is tightly controlled by nuclear pores. And so these are guarded by the nuclear pore complex, which helps to regulate any kind of molecule moving in and out. So nucleoli, these are dark staining within the nucleus, and this is the site essentially of ribosomal RNA synthesis and ribosome subunit assembly. Associated with the nuclear organizer regions that have the DNA coding for rRNA, these genes are very important for establishing phylogenies because rRNA genes tend to be highly conserved. And usually you'll see one or two of these nucleoli per cell. Chromatin. So chromatin is the assembly of DNA and proteins. It is sort of the packed form of DNA. 30% negatively charged DNA, 60% positively charged proteins, and then you'll have the 10% RNA associated. And so it's arranged in functional units called nucleosomes. So DNA wrapped around histones. This is where you get differential packing due to either acetylation, things like euchromatin, heterochromatin packing, and so that helps to regulate gene expression. If you're very tightly packed, there's no room for the transcription machinery to get in there and actually transcribe your DNA, so it's not likely to be expressed. Chromosomes, for instance, are composed of this condensed chromatin, and this condensed state helps to protect it and of course allows us to store it better, just because we have such an incredible amount of DNA. So moving on to the cell cycle. So a cell undergoes changes from the time it is formed until it is reproduced. And there are two major periods of the cell cycle, but one of them is like 90% of it. Interphase is occurring for 90% of a cell life. This is just when the cell is reaching maturity and then doing its job until it's ready to reproduce. When it's ready to divide, it enters into the mitotic phase and will divide into two. That is a very short part of the cell's life cycle, like only 10%. So interphase, cell's just out there doing its job. The nuclear material will be in an uncondensed chromatin state. That way it can be expressed. And it has subphases, including DNA replication. So we break these down into different phases. G1 or GAP1, this is growth and metabolism. If it's ceasing dividing, as in it's already reached its size, adulthood, and all the real estate's taken up, we call that G0 phase. And so there's no room left to divide. The cells are just simply functioning. In the S phase, the synthetic phase, This is where you'll actually get your DNA copied, so DNA replication occurs. And in G2, now we're preparing to divide that copied DNA into another cell. So good representation of that. In DNA replication, prior to division, the cell will make a copy of DNA. The DNA helices will be unwound and unzipped. The replication fork is the point at which it separates. The bubble is the active area of replication. And then you have these each strand acting as a template for new complementary strands. This is actually started off by an RNA primer. Now the thing is, you can't actually keep that primer. Remember, RNA has a different sugar and it uses a different base. It uses uracil. DNA doesn't like when uracil is incorporated. So you're going to have to get rid of that primer somehow, and that actually leads to problems down the line for us. But DNA polymerase will attach to the primer. It will incorporate nucleotides from solution complementary to the strand. And it will synthesize both strands at one time. We call one the leading strand and one the lagging strand. But DNA polymerase only works well in one direction. So one strand, the leading strand, will be synthesized continuously, but the lagging strand is backwards. So it is synthesized in segments or fragments and then pasted together essentially. So DNA ligase will splice those short segments of the lagging strand together. So what you're left with then is two identical DNA molecules formed from the original. This process is semi-conservative, which means that the template strand is actually left in the helix with this new strand. And so this is what that looks like. You can see that the helicase has already split the DNA, broke all the hydrogen bonds between here. Now we're in our replication bubble. DNA polymerase is synthesizing the leading strand continuously, but the lagging strand in Okazaki fragments that later have to be pasted back together with DNA ligase. So most cells are replicating and growing continuously. Not all. For instance, skeletal, cardiac, and nerve cells do not divide efficiently, and they do not replace themselves efficiently. If you damage them, you will get scar tissue there instead. Moving into M phase then, we have two events going on, mitosis and cytokinesis. Now this process has to be very tightly controlled because there is a word for uncontrolled cellular growth and division. That is cancer. Now cancer also has the benefit of metastasis, but any type of uncontrolled cell growth is not good. So mitosis in its simplest form is the nuclear division. So the DNA is duplicated and distributed to new daughter cells. There are 4 or 5 stages depending on who you talk to. Some like to include a pro-metaphase in here as well, but we want to make sure that each cell is receiving an identical copy. So in prophase, we have early prophase where the chromatin condenses and will become visible. Each chromosome has a sister chromosome, a chromatid, and they're held together by a centromere. The centrosome begins synthesizing the microtubules and push the centrosome to the opposite side of the poles, called the mitotic spindle. During late prophase, the nuclear envelope will start to fragment up. The microtubules will attach to the kinetochore and pull the chromosomes to the center of the cell. Once they reach the center, we form a metaphase plate. And so this is sort of an arbitrary time point. Basically, this just means that the chromosomes are precisely aligned at the cell's equator, the metaphase plate. And now the centromeres of the chromosomes will split, and each sister chromatin will become a separate chromosome. They are attached by these microtubules at the kinetochore and will begin to be pulled towards the poles, one chromosome of each of the original pair going to the pole. Then we begin telophase, which is almost the exact opposite of prophase. So once the chromosomes arrive at the poles, the chromosomes will decondense, so they will uncoil, the nuclear membrane will reform, the nucleoli will reappear, and the spindle, the mitotic spindle, will disappear as well. And then now we need to split up the cytoplasm and the organelles, and this is what happens in cytokinesis. So it actually starts far earlier in anaphase and then will continue throughout, but basically actin microfilaments contract to form this cleavage furrow, which will pinch the cells apart. So control of cell division is highly dependent upon signaling. And so GO signals include if your surface to volume ratio of the cell is getting too large, that means you're starting to reach the point of diminishing returns on your size because volume is cubic, surface area is squared. And so you're really diffusion limited here. Stop signals include the availability of space, and when they come into contact with other cells, it's kind of a good indicator that there's no more room to divide. This is called contact inhibition. Two groups of proteins are crucial for the ability to accomplish S phase and enter mitosis. Those are the cyclins in the cyclin-dependent kinases. So cyclins accumulate during interphase, and then CDKs activate cyclins when they bind them. In turn, they activate enzymes that prepare the cell for division. After mitosis, they're destroyed and the process begins anew. So this is necessary for maintaining the checkpoints. These checkpoints basically check if the copied DNA is actually of the correct fidelity. The G1 checkpoint is the most important of the three. If it does not pass, it enters G0, where no further division will occur. Okay, moving on to protein synthesis. So as mentioned before, DNA is the hereditary molecule. It is holding the code for all proteins to be encoded for, and it does this by directing the order of amino acids in a polypeptide. We call these coding sequences genes. Genes usually code for polypeptides. They do code for other things as well, but polypeptides is the main thing. And it's determined by the sequence of the nitrogenous bases here, adenine, guanine, thymine, and cytosine. And these form a triplet code, a codon, that will then code for an amino acid. For example, GGC codes for proline, whereas GCC codes for arginine. So the triplet specifies the amino acid. Genes also have exons and introns, in animal genes at least, in eukaryotes. Prokaryotes lack introns entirely, but introns are intervening sequences that allow you to alternatively splice one gene into multiple gene products, but you have to remove them in order to make a sensible product. So it's only the exons that actually code for the amino acids of a protein. The introns in this instance would be a non-coding segment just interspersed within that need to be removed. Okay, the role of RNA. RNA is an intermediate, essentially, in this instance. It is transcribing the DNA and then having that translated to protein. All RNA is formed in the nucleus, and it differs from DNA in that it is using uracil, of course, instead of thymine, and has a slightly different sugar that has an extra oxygen. Three main types of RNA would be messenger RNA, ribosomal RNA, and transfer RNA. Messenger is single-stranded. It is made from the DNA template and is complementary, except of course it is using RNA bases. And so this is referred to as transcription because we're not actually changing the chemical language. We're still going from nucleic acid to nucleic acid. mRNA is maintaining then this codon from the DNA. Ribosomal RNA is a little different, and this is one of the rare things that there are genes for that are not protein. There are rRNA genes. It is the structural component of ribosomes, and so this is the organelle, of course, where protein synthesis occurs. And basically, it's helping to translate the message from mRNA. Transfer RNA or tRNAs are carriers. They are charged with carrying an amino acid. They have a specific triplet code embedded in them called an anticodon, allowing them to carry only one specific amino acid. And so it will complementarily base pair with the codon of the mRNA at the ribosome, adding that specific amino acid to the growing polypeptide chain in a process referred to as translation. So translation, we have actually changed the language here from nucleic acid to polypeptide, or amino acid in this case. So protein synthesis occurs in two steps. First, we need to take our hereditary DNA. We need to transcribe that code into messenger RNA. Then we need to take that messenger RNA and assemble the growing polypeptide chain from it in translation. So continuing on with transcription, we're taking our DNA gene base sequence and turning it into a complementary sequence of mRNA. This process starts with transcription factors, which is actually a complicated protein complex that loosen the histones from DNA to open it up, basically exposing the DNA. They bind special sequences of the gene called the promoter, and then upon that binding, RNA polymerase will be recruited and will come in and synthesize the mRNA at the promoter region and into the gene itself. So transcription is broken down in three phases: initiation, elongation, and termination. During initiation, RNA polymerase is separating the DNA strands, so no helicase necessary. RNA polymerase can do it. Then it will incorporate the complementary RNA nucleotides to the growing mRNA matching sequence based on the DNA template strand. And what you're left with is a DNA-RNA hybrid where the two are temporarily bonded. Then during termination, transcription will stop when RNA polymerase reaches a special terminator sequence, a stop sequence essentially. So this is occurring in the nucleus. Before it can leave the nucleus, the mRNA transcript needs to be processed. So it is edited. Before then is referred to as pre-mRNA. You need to remove the introns. So the RNA polymerase doesn't actually know what's an exon and what's an intron. It just codes everything. It makes a transcript of everything. Well, you need to remove those introns with special proteins called the spliceosome, leaving only the exon coding regions. And so now going, so the process involves mRNA, the genetic code, tRNA and ribosomes, the translating events, and sometimes the rough ER, depending on whether or not your protein actually needs modification. So the genetic code, this is referring to essentially the chart that shows you how triplet codons get actually translated to amino acids. Remember that every 3 bases is a codon. Those get translated to messenger RNA, and the signal is essentially kept now in RNA form. There are 64 possible codons. These are universal largely. Mitochondria uses a slightly different scheme, but based on the 4 bases of RNA, A, U, C, G, and 3 places, so 4 to the 3rd equals 64. 3 of these are stop codons, so they don't actually code for anything, and when translation reaches a stop codon, nothing will be incorporated, and then some other molecule will come in and bind and disassemble the complex. The rest all code for amino acids. There are only 20 possible amino acids, so that means that some are represented by more than one codon. And this is actually important. The redundancy here helps to protect against transcription errors. So you've made an error in the third base. Well, if it codes for the same thing, that doesn't actually matter because you're still going to incorporate the same amino acid. And so here is our codon chart, and you can see of the 64 different possible codons, like 6 of them code for just leucine. So do you think that this is probably a really common mutation and that it's good to have a lot of redundancy here? It's probably somewhat true. But then you also have, of course, the 3 stop codons and our start codon AUG, which in eukaryotes codes for methionine, in prokaryotes codes for formylmethionine. Okay, moving on to translation, the role of tRNA. So tRNA will bind a specific amino acid based on its anticodon, forming an amino acid tRNA. The anticodon is the triplet that's determining which of those amino acids will actually form the amino acid tRNA, and then it will carry it to the codon on the mRNA that is complementary. The ribosomes coordinate the coupling of the mRNA and the tRNA, and the ribosome only contains 1 binding site for mRNA and 3 binding sites for tRNA. There is the A site, the P site, and the E site. The amino acid site for the incoming amino acid tRNA, the P site for that tRNA to link it to the growing polypeptide chain, and then the E site for once it's delivered its package to exit. So the sequence of events for translation is exactly what it is for transcription. You have initiation, elongation, and termination. So initiation, the small ribosomal subunits are going to be binding to an initiator tRNA, in this case coded for by the start codon AUG, making a methionine. The mRNA would be decoded after this. It will scan the mRNA looking for the first methionine, referred to as the start. So the large ribosomal subunit will then attach to the small ribosomal subunit, forming a functioning ribosome. And so at this point, the initiator tRNA is located in the P site of the ribosome, and the A and E sites are actually empty. Now we need to enter into elongation. So during codon recognition, the tRNA will bind the complementary codon in the A side of the ribosome, and ribosomal enzymes, this is very rare. Enzymes are biological catalysts and almost always protein-based. These are ribosomal-based, they are RNA-based. Will attach growing polypeptide chain from tRNA in the P site to the amino acid of the tRNA in the A site. So we're increasing the amino acids on our polypeptide chain and then translocating, so shifting the ribosome down 3 bases of mRNA, displacing the tRNAs by 1 position each. So the tRNA in the A site moves into the P site and then will deliver its amino acid there. The tRNA in the P site has already delivered its amino acid, so it will move into the E site and be ejected. The tRNA that's in the E site will be ejected from the ribosome. So once the A site is empty, the new tRNA enters, brings its cargo, and the process starts over. The mRNA is read, so an additional round of this will start. Polyribosomes are more common in prokaryotes, but it is multiple ribosome mRNA complexes producing multiple copies of the same protein. And they're able to get away with this because transcription and translation happen in the same place in prokaryotes. We don't have that luxury because we're transcribing in the nucleus and then sending it out for translation in the cytoplasm. But essentially what you can do is take one transcript and make multiple proteins from it. So termination then. So we need to be able to stop this. We don't want it to go on forever, otherwise we're just going to make a nonsense product. Well, you encounter one of the 3 stop codons, either UGA, UAA, or UAG. When the mRNA enters the A site there, translation is going to end. So protein release factors will actually bind there. It will cause water to be added to there instead of another tRNA. And this will lead to the disassembly of those ribosome subunits and the degradation of the mRNA product. And so after that, your polypeptide product might need a little help getting shaped into the correct 3D shape. Some proteins need chaperones, etc. But from there, the polypeptide will be released and allowed to fold up however it needs to. Okay, transcription. Continuing on, the role of the endoplasmic reticulum in protein synthesis. The endoplasmic reticulum has a signal sequence present on growing protein on polypeptide chains that can tell ribosomes to dock on the rough ER surface, and a signal recognition particle will direct the mRNA ribosome complex to dock here. And so once docked, it will synthesize the polypeptide into the ER, and as a result, that growing polypeptide chain can now be modified. It can have sugar groups added to it. It can be enclosed in a vesicle and then sent for transport to the Golgi apparatus. And so in summary, we got complementary base pairing directing transfer of genetic information, basically going from DNA to RNA to protein. We actually call this the central dogma of molecular biology. These DNA triplets are coded into mRNA codons. Codons are then base paired with tRNA anticodons. This just ensures that the correct amino acid is incorporated. And the only exception is, of course, there's no thymine in RNA, instead you use uracil. So other uses of DNA and RNA, well, DNA has structural genes for microRNA, ribose switches, small interfering RNAs, and even the rRNAs. So microRNA are small RNAs basically that can silence mRNAs if they bind them. So this leads to differential expression of genes. Ribose switches, these can control protein synthesis on or off depending on environmental conditions. Small interfering RNAs are similar to microRNAs, but they are used sort of as a rudimentary immune system to deactivate viral genes. And we've done some pretty interesting stuff with small interfering RNAs. For instance, if you have a purple flower, say, I really would like this flower to be more purple. I'm going to find out what gene that makes that purple pigment. I'm going to take an mRNA transcript of it. I'm going to inject it, and it's going to make more copies of that, right? Well, you do that, and now suddenly the flower turns completely white. It has deactivated those purple pigment genes. Why is that? Because you injected a small interfering RNA that told it to silence those genes, essentially. Okay, moving on to apoptosis, autophagy, and proteasomes. Autophagy, so cells that have become obsolete or damaged. Of course, you need to be able to do something with that. You can't just let them sit there. There are chemicals inside the cell that are very destructive. For instance, the contents of the mitochondria, the contents of the peroxisome and the lysosome. You can't let those just sit in there. So you need to be able to process and dispose of these non-functional organelles and cells by autophagosomes. And then, of course, you would fuse those with some type of digestive organ. One other way that you can deal with defunct proteins is by ubiquitin tagging. So if a protein is misfolded, or it will take the wrong shape and thus not be able to function. And so you can mark it for destruction by a ubiquitin tag. Tag it with this molecule ubiquitin, and the proteasome, which is kind of like the trash can of the cell, the protein trash can of the cell, will disassemble that protein and try to make use of the amino acids. So apoptosis is programmed cell death. It is just a natural course of living, essentially, but certain cells do not like to do that. Cancer cells, for instance. But it just causes them to self-destruct, hopefully without damaging anything nearby. As mentioned, the contents of the cell are actually quite destructive to neighboring macromolecules and stuff, but this starts with mitochondrial membranes leaking chemicals activating enzymes called caspases, which lead to degradation of macromolecules like DNA and like this in the cytoskeleton, actually, leading to cell death. As a result, the cell will shrink and be phagocytized by macrophages. So, developmental aspects. So it's hard to really imagine this, but all somatic cells share the same DNA. They have exactly the same copies of everything. Every cell type can be induced into another cell type, but they do not carry out the same function. So how do you keep the cells of your eyes, for instance, from synthesizing hydrochloric acid like the cells in your stomach do? Well, it all has to do with regulation and signaling very early on in development, telling the cell to develop along a certain pathway. And it does that by turning some genes on and others off. And this cell differentiation is incredibly important. Destruction and modified rates of cell division. So well before we are functionally born, essentially our organs are already in place, and now we just need cell division for growth, essentially. In adults, all we need cell division for really is to replace the cells that we're already using and to help repair any damage that has occurred. In accelerated growth, we would call that hyperplasia. So an increase in cell numbers. In atrophy, we would call that a decrease in size resulting from loss of stimulation or use. And so atrophy is just a normal consequence of aging, but is unfortunately something we'll all have to deal with. So we're not exactly sure on the exact prescribed causes of aging, but there is wear and tear, of course, a lifetime of exposure to harmful chemicals and free radicals that just occur naturally as a result of metabolism have cumulative effects. Free radicals coming from the mitochondria diminish energy production. Immune system disorders, so in kind of autoimmune response, your body not being able to recognize self. The progressive weakening of the immune response as well. So in animals, for instance, one of our immune organs is the thymus. It's predominant at birth, and then as you age, it diminishes and atrophies, and you start to get sick. There's also the end replication problem I alluded to earlier, where RNA primers being used to start off DNA replication isn't great, because when those primers are removed, they're recognized as single-stranded breaks and repaired. You can't do that. When you repair them, they're just kind of clipping the end off. But every time you enter a replication cycle and you just clip an end off an exon like that, that's bad. And so this end replication problem is mediated by an enzyme called telomerase that's supposed to help protect us, but deeply implicated in the aging process as well. So these telomeres, telomeres are strings of nucleotides supposedly that protect the ends of the chromosomes. They're just nonsense repeats at the very ends of the chromosomes that are supposed to protect us by shortening, acting like essentially a buffer against this end replication problem. And so telomerase is the enzyme that lengthens them. So found in germ cells of embryos but absent in adult cells, except for cancer cells notably. So this is why we do not simply upregulate our telomerase, because we associate this upregulated telomerase with uncontrolled cell growth. Telomerase is actually what makes immortal cell lines, like those cell lines of HeLa cells, Henrietta Lacks. Anyway, I know this was a long section. Please break it up into the divisions that you need to, but keep in mind this will be on the Module 1 exam. Thank you. Have a good day.